So, good evening, everyone. Thank you for joining us for one more of these special curated sessions on subjects which we will be teaching at Sai University, but in alliance with other fields. So, the idea is to project the interdisciplinarity nature of the way we teach, regardless of the field. So, today we have with us Dr. Toleti Subba, Professor of School of Arts and Sciences. So, brief introduction. Dr. Subba has 34 years of academic experience in microbiology, biotechnology, and biological sciences. He has worked across the domains which include microbial ecology, environmental biotechnology, industrial cooling systems, and environmental impact assessment. Dr. Rao earned his PhD in biotechnology in 1999 from Anna University and the degree of Doctor of Science in 2020 in biotechnology from Periyar University. He has received many awards, such as the International Meritorious Contribution Award 2019 from the National Association of Corrosion Engineers, or short form NACE USA, from the Department of Atomic Energy Group Leader Award 2018, SK Seshadri National Award 2013 from Electrochemical Society India, Indian Institute of Sciences, DAE Group Achievement Award 2011, and NACE International Excellence Award 2006. He is a fellow of the Academy of Sciences, Applied Biotechnology Society, and a Senate member of Aragappa University and member of the Board of Studies at several universities. He has over 115 journal publications and published several classified technical reports for the Department of Atomic Energy. He began his professional career as technical officer with Indian Veterinary Research Institute, UP, Uttar Pradesh, in 1987. Subsequently, he joined Bhabha Atomic Research Center in 1988, Kalpakkam, as a scientist and was throughout associated with it before joining Sai University. In his career span, he has been associated with many national institutes and universities, such as the faculty at Homi Bhabha National Institute, and collaborated in various projects with the National Institute of Ocean Technology, Indian Institute of Science, Nuclear Power Corporation of India, Aragappa, Pondicherry, Sri Ramachandra University. At Sai University, Dr. Rao will be engaged in teaching and research in his area of expertise, as well as will be responsible for setting up the biology laboratory. So over to you, sir. You can.
Good evening, dear students. It's a pleasure interacting with you on the expertise on my own interest in biological sciences. So I have structured the talk in such a way that it will be easily understandable, and also I would like to describe the transdisciplinarity science aspects of biology and then its interactions with other subjects of science and technology. So now let me begin with the arts and science relation. So if you look at CERN, CERN is the European Council for Nuclear Research and has the mandate of world-class fundamental physics research organization. So it invites liberal artists to spend time at CERN to facilitate close collaborations with scientists and to understand and represent the structure of the universe. This is done because it has to motivate the scientists working there for better understanding the nature, life, and universe. So with this small intro, I thought of taking you into the Thomson's landmark book on growth and forms, and which describes the theory of transformation. So according to Thomson, he says that one species evolves into another not by successive minor changes in individual body parts, but by large-scale transformations involved in the body as a whole. So this book, I suggest those students who are interested in biology to read it. It is available online as an ebook. And then this gives you the organic magnitude and the rate of growth of cellular forms and structures, adsorption in the forms of tissues, and then examines a vast spectrum of life forms. Likewise, if you look at the intersections of arts and science through time and paths, one good example is the how art and science intermingled based on observations and interpretations. The architectural marvel called La Sagrada Familia, which Antoni Gaudí designed and built in Barcelona, Spain. He was inspired by the geometrical features found in nature, like, for example, branching of trees and then staircases with seashells like curves, and designed the physical structure of the church. So this is nothing but it can be described as biomimetics. As of now, biomimetics is playing a major role in science and technology. For example, if you look at the Boeing's surface finish of the aircrafts, they have been designed based upon this shark skin so that it reduces the drag, air drag, and then also brings in 15% fuel efficiency. So this is one example of applying this biomimetic technology. In my experience at Kalpakam, also we have used these biomimetic techniques. Like, for example, uh, the seashells, which you can see along the seashore, which are of the bivalve organisms. They have a particular pattern on the or morphological or topological features which will not allow anything to grow on that surface. So this is what we wanted, because this is they have what is called a rippling property, and this is we have imprinted the surface topology onto a polymeric matrix and tested it by exposing it into water and see whether any life which is present in the water is sticking to these morphological features or the uh, topographical structures, such that there will be prevention of these growth on these structures, which are some useful in applications, which I will describe you in my talk later. So now, similarly, another area of interest is the studies on the structure and connectivity of the brain. So Daniele Bassat, a neuroscientist, viewed a multidimensional network data with an artistic lens by enlisting artists and designers to help brain connectivity. So this has inspired and enabled cutting-edge technologies such as the virtual reality and rapid prototyping, which are accessible and inexpensive techniques which are being widely used in biomedical applications. Similarly, if you look at biological sciences, it encompasses all the divisions of natural sciences, examining various aspects of vital processes. And the concepts include anatomy, physiology, cell biology, biochemistry, biophysics, organisms, animals, and plants. So biology studies everything living on Earth, and from the simplest and the smallest single-celled organism to the complexity of human brain. The science of biology also influences everything on planet Earth, which I will be able to demonstrate during the course of this talk. If you look at this image, it describes the sizes of the atom on towards the blue whale size. That's to depict the different morphological sizes of the life. So this slide, which is on life science cycle, describes you how from the molecule, it is the biochemical constant of a cell, an organelle is formed, which is a specialized subunit in a cell, and which results into a cell, which is a functional unit of a living organism, which collectively transforms the tissue, and then the organ and the organ system, and finally the organism, a single individual organism. And these will together form the population, and then also the community, and then which interacts with the ecosystem, and finally it is the biosphere, the world we live. This is the life science cycle, which I think all of you are going to experience or familiar with. So coming to the interest in biological sciences, I thought of sharing with you some important discoveries which have revolutionized life sciences, which like, for example, it began with there is some, yeah, the first one actually is not able to highlight it here because it is on the Darwin's theory of natural selection, which says that the fittest will survive. And coming to the next major discovery is the antibiotics. Even today, you might have been listening to that the new antibiotics are being discovered and being tested and being released to the market for human applications. So the Alexander Fleming's penicillin is the primary antibiotic which there have been many synthetic antibiotics which have been uh, developed and then applied for humans. Similarly, if you look at the another important discovery is the HeLa cells, which is of great significance to human research in science and technology. It was commonly used human cell line for all the biological studies. The line was derived from the cervical cancer cells taken from Henrietta Lacks. That's why the name HeLa, who died of cancer. And now these cell lines represent scientists' first opportunity to do many human tissue studies, which took advantage in a variety of things from disease prevention to space physiology. So if you look at this image, it shows the HeLa cells, which are stained with the fluorescent hex dye, which turns their nuclei into blue color. This is the specific dye which will be staining only with the nuclei of these HeLa cells. Similarly, if you look at the another major discovery is the DNA helix, and then all of you might have studied in your school days about the DNA structure and all, and it's has revolutionized the recombinant DNA technology and as well as resulted in the development of molecular biology science, which is now playing a major role, which I elaborate in my slides later. Likewise, the E. coli transformation by incorporating a plasmid or a gene of interest into it, and this has resulted in the that recombinant DNA technology or the biological transformation of organisms which are of specific interest to human applications. Likewise, the 1983 polymerase chain reaction discovery and then its application has almost revolutionized molecular biology science, and then also on the detection as well as the its application in many aspects of human activity. It is nothing but it will amplify, copy small segments of DNA, and then gives us the required nucleic acid content for further analysis or either in identification or in detection. Likewise, the green fluorescent marker protein, which was being isolated from a jellyfish, which also has revolutionized biological research. And this you can see in some of the images later, which I show you how this green fluorescent protein gene is incorporated, which is the result of a particular gene expression like that. Likewise, you have many fluorescent markers which have revolutionized the research in biology, which are the EBFP stain for the nucleolus, and then for mitochondria, and for vimentin for endoplasmic reticulum, for the nucleus, as well as for the Golgi apparatus. So now, if you would like to see a cell with all of its organelles, you have today fluorescent stains which can be applied, and then you need to have a fluorescent microscope to observe them in a and for as far as for the research as well as for the educational purpose. So these are the few discoveries which I thought of sharing with you, which have almost created dynamic research activities into the biology for human applications. Likewise, Dolly. Dolly is the sheep which was being created using the somatic cells from the mammary glands by just the process of nuclear transfer. So after its birth, it has grown, matured, and reproduced naturally. This image shows the sheep dolly and her surrogate mother. This has almost revolutionized again the research in biology, and now pigs, deer, horse, and bulls have been cloned using this technique for their advanced yields in milk as well as in meat. Uh, Likewise, also scientists cloned extinct animals by collecting nuclear material from the frozen tissues. This is for of academic interest, and then and these are having limitations as far as ethical applications are concerned. You might have seen the Jurassic Park movie, which is one of the uh, applications, which means on the fiction stories which have been generated as part of this activity. Similarly, stem cell research. All of you know that stem cells are the one which will help in the development stages of a human embryo. And then the scientists from Coyote University and Wisconsin reverted the adult skin cells so that they can act as with pluripotent stem cells. That means they have the capacity to transform or develop or differentiate among themselves into different types of things. Like, for example, you can create kidney tissue, likewise liver tissue, heart tissue, etc. And particularly this has resulted in the applications of skin burn treatments, wherein the skin of the burnt patient can be generated using these stem cells, which can be transformed from any of the somatic cells of the body. So now coming to the genomics and proteomics, you might have heard these names, but genomics is nothing but study of organisms' genome and the use of these genes, and it deals with the systematic use of genetic information and then with other data to provide answers in biology, medicine, and industry. While a genome is a constant entity, a proteome is the one which is more complicated than the genome, and because of its differences from cell to cell, and is constantly changing its biochemical interaction with the genome and the environment. So proteomics is more complex, and which has now almost as wide applications in pharmaceuticals and in industrial applications in generating many useful human application products. Similarly, meta-barcoding is the one which is now being used. You might have heard about classification of plants and animals. Earlier, conventional classification experts were using morphological features as well as some metabolic indicators as the tools or the markers for identification of organisms. But now that has completely been lost, or many of those conventional taxonomies have not shown much of interest, and it has resulted into development of this barcoding way of identification of organisms, which is based most commonly based on the 16S rRNA gene in bacteria and 18S in eukaryotes and cytochrome oxidase 1 in metazoans. Metazoans are nothing but zooplankton which live in marine environment or in aquatic environments. Like, for example, you have a complex growth of biological organisms on a surface, which we term it as biofouling. It will have a few tens of organisms. The whole DNA of this can be extracted and amplified using PCR, and then can be separated and for identification, and can be sequenced using this Illumina sequencer. Now, the advantage is that we can do large-scale taxonomic identification of complex environmental samples using these DNA sequences, which are result of a short regions of one or a few genes. And these are called as DNA barcodes, which are specific for the organisms. So this kind of digital microbial or organism library is being developed at various institutions. So, and it is freely available and shared among researchers for identification. So this also has helped in large-scale analysis of environmental samples. Like, for example, you collect soil samples, water samples for a characterization of the organisms present in that. You can use simply extract the DNA and then go ahead with this DNA barcoding procedure. And then it's, although the technique is very expensive, but as of now, with development of new methodologies and procedures, it is becoming cheaper. Now, another interesting subject of biology is on the bioinformatics, which is a subdiscipline of biology and computer science. With the acquisition, storage, analysis, and dissemination of biological data, mostly the DNA and then amino acid sequences. You may be understanding by now that the human genome is completely being sequenced, which has 3.3 billion base pairs. Now, what does this mean? We have so many of these gene pairs, but what does this mean is still the information is not being cracked. So bioinformatics is science which provides the tools which are interlinked using various subjects, which also such as computer science, chemistry, biochemistry, biology, statistics, mathematics, and engineering, to understand the applications which have to be derived from this presence of genes, as well as also the base pair interactions. Likewise, computational biology, which also plays a major role now, where biology and mathematics together are intertwined. The advent of this genome sequencing, functional genomics, systems biology have almost revolutionized the application of algorithms from statistics and machine learning, which are applied to the genome studies and to classification. And now this is the science which is been developing slowly, which is still young, and it has a lot of promise. Like digital biology is an emerging and promising science as far as the new science and technology developments is concerned. If you look at this biomolecular structure, machine learning is transforming our understanding of protein folding. So now you can transform or also modify these protein structures in such a way that which particular structure or a function will have its own property of a particular biomolecule. And then fluxonomics. This is nothing but the approach to seek to determine the rates of metabolic reactions which are happening in a biological cell or a biological entity. Like, for example, you have cloned a particular gene of a particular protein, like say, for example, insulin gene has been cloned into bacteria and it is grown in a fermenter. And you should see that the insulin release from this bacterial cell is following this curve. And after a particular time of the growth of the cell, it comes to stationary phase. So you need to extract that insulin between these two phases of between this log phase or before entering into the stationary phase for the downstream processing of the protein, which is of like insulin, which is of importance to human applications. Likewise, now you might have heard about the word of in vitro, in vivo. In vitro means you do it in test tubes. In vivo is inside the animal. But now in silico analysis has becoming popular because this is based on biological and physical knowledge and data discovery, wherein you collect this information and then use experimental design to test an hypothesis. So these will variety of software programs which are available, which will nothing but having the information to be fed in the form of amino acid sequence or the gene base pair interactions. So this will result in models of gene regulation, biochemical networks, cells, and organs. So this way, what you do is you run a dry experiment and analyze and test the hypothesis which you are of interest and which you are interested. So up to this stage, there is no need of using except only a good computing system as well as the data available in a particular of the particular genome or a particular protein. After this, you will develop experimental techniques to further confirm or to further test your hypothesis based upon these computational dry runs. And then finally, you do the wet experiments to verify or reject your hypothesis. So now it has come to such a stage that you can able to design, like earlier, there used to be some failures in experimentations, but using this in silico analysis, you can be so sure that you can go up to the level of your interest of application and then design your experiments, test the hypothesis, and then do the wet experiments and get the good results which are having human applications. Now, another interesting thing which people will be amazed to know is about 3D printing of organs and tissues. So you might have heard about 3D printers. 3D printers in industrial application is already been in practice for the last 5 to 6 years, and it is entering in the Indian market also. Whereas 3D printing of organs has not yet been started in Indian science and technology. It's in any organ or a tissue can be printed using a particular tissue engineering procedure. So, like for example, based upon the interaction and the training skill and the education, you can also have a result into cell reactions, cell therapy, drug screening, tissue regeneration, and organ transplantation. A human heart has also been printed using this technique. And then, particularly in accidental scenarios where people lose the ear or nose, there have been printed and they can be applied in such a 3D printed self-engineered cells results in no rejection of the graft which you are doing. You might have heard about kidney transplant and all from donors. In such cases, no, the patient will be given immunosuppressive drugs such that the kidney is not rejected. And these immunosuppressive drugs have to be taken throughout the life. And this will keep the patient in a risk form wherein you should not be exposed to any disease. Like, for example, you might have heard about COVID outbreak, and such patients are very risky to because their immunity levels will be very, very less. So now with the advent of this 3D printing of organs, the biology has completely revolutionized the treatment and means the cures and then the treatment of multiple accidental scenarios of human applications. Now, I'm talking about only 3D printing, which has still not entered into the Indian science and technology, but 4D printing, 5D printing have already been established in Europe and United States. So 4D printing is nothing but it's almost like a similar 3D printing thing. Only the final influence will be from the input of a particular factor. Like for example, in this case, a temperature is one factor which transforms the 3D printed product into the applicable product. Similarly, 5D printing is nothing but the printable object will have 5 degrees of freedom in demonstrating its application. So these two are of now major industrial applications only. Still, it has not entered into the human application or human organ printing as well as tissue engineering aspects. So there's a lot of promise as far as biology research is concerned, and it will be quite investigative as well as quite interesting for the students to learn about these things. And another is synthetic biology. Now you might have heard about bacteria, viruses, plants, animals, etc. So these are all been evolved on development, human development on the developmental scale, whereas synthetic biology is the one which you synthesize. Like, for example, use all these techniques. Like, for example, you can use bioinformatics and computer science tools to know that, for example, insulin is produced, and then insulin is made up of 51 amino acids, whereas the required active amino acid sequence probably can be 31 amino acids itself. So if you know about that information, you can use that 31 amino acids sequence and then engineer it, and then synthetically, using synthetic chemistry, you can develop them and then put it into a microbial organism, do molecular analysis, and finally see whether you are going to get a similar insulin, like a functional insulin product. So this is nothing but synthetic biology is going to minimize the required genomic information, so that when you get the final product, you will get only the final product. You will not get any other metabolic products which will interfere in your downstream processing of a particular product or a compound of human application. So this has just begun, and it is being applied in few areas, and a lot of promise is there in synthetic biology. It will be using biological modules as the components with which one can engineer new biological systems which are having a minimal genomic sequence, and then now make large scale genetic engineering easier and more predictable in getting the product. So it has novel industrial and therapeutic applications, but the science is still evolving and developing. So this is one of the interesting things which also inspired me, like that we can use and develop new microbial systems for application for both, most particularly for industry as well as for human applications. Now, with this, I would like to share a little bit on my earlier experience at Department of Atomic Energy, where I worked. And then, if you look at the Department of Atomic Energy's program, it has three phases. One is the stage one is the pressurized water reactors, which are about 22 in operation now, which generate close to about 25,000 megawatt of electricity. The second stage is still in the trials, and 500 megawatt plant is coming at Kalpakkam. And then the third stage is the one where thorium will be used. So with this second stage demonstration, we will come to the energy self-sustained system in India. So now we look at why nuclear energy means energy security is national security. Now the technology is so mature that it is also competitive with other forms of energy, electricity production. There will be no climate change releases, and then it is having a proven safety record for the past 60, 70 years. Reactors are in operation, and it is a concentrated form of energy. The only operations are on the safety of the reactors and the nuclear waste storage. Now, in both these areas, India has matured technology and research, and we are almost becoming leaders in this area. Now, if you look at a typical power plant, which will say this is the box, and then Here, nothing is bad that you generate steam and steam will turn the turbine and then electricity is generated using a dynamo system. So now, for example, the Madras Atomic Power Station, which I will describe a little earlier, it takes seawater from a tunnel and then goes to the pump house. And then this is nothing but the intake phase of the water, and it cools the condenser. When it condenses the steam, and then this is the plant phase, and it comes out and is discharged through a canal and joins the sea. So this is the plume phase. That means the water which is taken inside will be about 25 degrees, and it cools the steam and it goes out as heated water, which is having about 35 degrees. Means a 10 degrees increase in the water. And same way, we will add chlorine, like we use chlorine for disinfecting the drinking water. Here also chlorine is used to kill organisms from entering into the system. So it means the organism or the life in the water will be having a chemical stress because of chlorine, and a mechanical stress because of its pumping into the pipelines and other structures, and finally the thermal stress because of the increase in 10 degrees. So these are the factors which will be playing on biology. This is what I was studying at or researching at Kulpakam. So this is the artist diagram of Madras Atomic Power Station. It has two reactors, and then seawater is drawn through a subseabed tunnel. This bridge is about from here to this end, it is about half a kilometer into the sea. Here, this is the well through which the water enters and comes to the pumping station and goes into the condenser system. The heated water is released and goes back and joins the sea. So this is the bridge, and this is the well, and this is the tunnel through which the water is drawn. So up to this depth, it is 13.5 meters of seawater depth. And from here, the tunnel has been drilled. So this is about minus 50 meters. And it should be in a rocky area. And then finally, it comes to the plant area where from here the water is used into the condenser system. When such water is drawn, along with this, whatever life in the water also comes in. So this has results in the growth of these different types of organisms on different materials which are of interest in the industry. And then this is how to control that. Now, finally, we also did some work on how we can use these organisms for better human applications. Like, for example, the bacteria which are growing on those surfaces, which I showed you in the previous slide, you can see that they can be transformed into granules. Means these are the granules, and these granules are having GFP protein. That's why the green fluorescent color you can see here, which I told you in my initial biological discoveries. And these granules can be grown using only one tank. So just using one tank, you just fill it up, you go on mixing it, and then the reaction will create these granules. You stop it, the granules will settle, and then you get a clear water or effluent, which is a treatment. So these granules, which have a shelf life of about 3 months, you can store them for any applications. Like, for example, we have used this technology to treat our own Kalpakam Township sewage. We have two plants which can treat about 250 hours sewage generation, and similarly 1.5 million liters per day plant also has been erected, which treats about 2,500 houses. So this way, we are using whatever the technology which we have developed for controlling this growth in industries to societal application. Likewise, we are also now working on developing phototrophic granules, because the earlier granules which I showed you, we need to provide some energy source for their growth. Whereas in these phototrophic granules, we have microalgae and cyanobacteria, which will capture sunlight and carbon dioxide, and then they are self-sustained. So this is what we want. So these are the reactors in which we can grow these granules, and which can also treat the raw sewage. And you can see this raw sewage here, and then this is the clear water after treatment in the granules have settled. The advantage in this process is that during the photosynthetic process, the pH of this water will go up to 10 to 11. So that increase in the pH kills the pathogens, and the water which is means the wastewater which is been treated by this process can directly be used to gardening and other toilet flushing purposes. So this image shows the different shapes of the phototrophic granules which we have developed for applications, and both for industry as well as for societal applications. So this is the images which show you the various stages of this granular development. So by 70 days, you'll get a very good granules, which you can easily grow them in this kind of bioreactors. So finally, also I would like to share with you the the question that radiation is a issue in a nuclear power plant and all of that. So we want to use some bacteria which are radiation resistant. So for example, a bacterium can grow up to 15,000 gray. Gray is a unit of radiation, and without much damage to it. So it happily grows in radiation environment. So this bacterium, we want to use it as part of the cleaning of nuclear waste. And then this images, this is an atomic force microscope image of this bacterium, which is named as Deinococcus. And this bacterium is incorporated with the GFP gene, which means that the green fluorescent protein, which I earlier talked to you, you can, so that if you can put it in a fluorescence microscope, you can easily see without any staining is not required for these things. And these are vital applications in nuclear waste treatment. Likewise, coming to environmental impact studies, at Kudankulam, you might have heard about that Russians are helping us in building up very big reactors. There are 2 reactors which generate 2,000 megawatt. So we also want to know, when mega power projects have been installed in the near coastal regions, what is the impact on the operation on the coastal waters. So we also do a lot of studies on the distribution of this water which has been released, heated water, and you can see those images here. Based upon the sea current, the heated effluent current moves in different directions in different times of the year. And this data is of vital importance because based on this dilution factor only, we will be able to tell up to what area this industrial operation is impacting the environment. So there are regulations which have been put by the government. That means the difference of the water which you are taking from inside to a plant and released, it should not be more than 5 degrees. For old plants, it is 10 degrees, but now it is 5 degrees. So with this, we are able to monitor the impacts on the environment and also recommend to the plants, operating plants, to take suitable measures. Another interesting area which is now Government of India wants is the deep ocean mission, where India wants to map the deep oceans of both Bay of Bengal, Arabian Sea, and Indian Ocean. So among them, sea mounts are of great interest, because on these sea mounts are the extinct volcanoes, and which rise from the sea floor to about 4,000 meters. So research has to be initiated in this area, and also on the type of deep sea organisms to be recorded. And similarly, deep sea environments are also famous for these manganese nodules, which are generated by biological process. These are pure manganese nodules. Like this, there can be of a variety of metals which are purely been there that needs to be mined. So the deep sea mining is another area which is going a lot of promise for these vital rare earth elements such as cobalt, platinum, and molybdenum. You might have also been heard about atmospheric carbon dioxide increase, global warming and all. So if you see this graph, it shows that up to 1950, the levels of carbon dioxide are below 300. And whereas within 60 to 70 years, we have increased up to more than 430 ppm. So this is alarming because this is all because of human activity only. So this is 60% is from the fossil fuel burning. So if we stop fossil fuels and then bring in renewable energy and then nuclear power, we can prevent this. Otherwise, this carbon dioxide production levels, which are increasing, may cause climate change and also result in what is called as ocean acidification. Is an important new area which is affecting the life in sea, because all the shells which are being synthesized by the organisms are made up of calcium carbonate, and if more and more CO2 is getting dissolved in seawater, this carbonate ions presence will be reduced, and as a result, less of carbonate is available for the organisms to pick up from the water and then make these shells. Likewise, from 1960 to 2021, the CO2 emissions were increased, and from the analysis of 189 countries, it is found that a 1% increase in the GDP is associated with 0.5 to 0.8% increase in the CO2 emissions. So that means if you want a good economic growth, you will be using your resources, and then also you are going to destroy your biodiversity. So there's a lot of applications of biology in economics aspects also, as well as for the survival of humans. So this is what I thought of sharing with you as part of the biology promise in future. And as everybody is telling, 21st century is the century for biological sciences. There are many wonderful discoveries to be invented, and then there are wide applications for human application. So with this, I will end the talk, and if you have any questions, please.
Thank you, Professor Rao, for that fascinating session. And honestly, even I didn't realize that biology, when we study in school, is so set as a means to either become a zoologist or you end up being a doctor, but that it has application in so many fields, including economics. I didn't think of it from that point. So that was really fascinating and enlightening. So participants, you can raise your hands, and if you wish to unmute and ask questions, you can go ahead. Or if you'd like to put it on the chat box, you can do that as well. So participants, feel free to ask. I know some of you are actually coming for biological sciences. I know by your profile. Why don't you ask your questions now? Any question? 1 second. I see some chat. Oh, yes. Okay. So we have a question. In one slide, it was said that scientists cloned extinct animals too. May I know any species cloned which reproduced and is still surviving?
Yeah. I explained to you that Dolly is the sheep which was cloned from the memory cells which were taken, and it has survived, and it is matured, and it also has delivered a normal sheep with time. So likewise, other animals which have been cloned also, the offsprings and the generations are being normally continuing.
Okay. How is 3D printing biotechnology going to help in the medical field?
Like I mentioned to you a few things, like for example, you have a major damage of your nose or a ear during an accident. Earlier, it is that with possible the skin from your thigh region or else have to be what's called as architected into your shape of your ear or a nose and then being stitched. But now with the advent of this 3D printing, your tissue will be grown and then printed to your shape of your nose based upon your earlier photographs. That is enough. Like, for example, it is nothing but again using CAD, computer-aided designing, and then synthesizing the particular requirement. And it is being proved very well in the American and European countries in many vital cases of accidents where people lost a major of most of their vital systems.
No, I don't think there are. So otherwise, by now we would have received a response. So there are no further questions, then yes, I think we can call it a wrap. Thank you so much, Professor.
Yeah.
And thank you, participants, for joining us. And we look forward to more such sessions in the coming weeks till you commence your program. If you have any specific questions, though, for biological sciences, feel free to route it to me. I can put that in the chat box.
Or you can email them. They can also email, and then we can also look at it.
Sorry, sir?
Yeah, if they have any questions, they can mail them to you or me, and then we can.
Yeah. Whichever is your preference, sir. If I can put your mail directly, it's fine, or I'll just route it through me is also fine. I'll put mine. Let them route it through me. I'll connect you to them then.
Yeah, it's okay.
We have one more question, I believe. Okay, no, it's just comment. Arifa says, thank you so much, sir. This webinar was really mind-blowing, mind-opening. So thank you, Arifa. And I think you should thank Professor Moritz.